Today we're going to talk about sequence-based microbial surveys. And so we've talked so far about how you identify an organism based on some molecular data, that sequencing out of the environment, collecting some sequences, and going back and identifying a specific creature. Today we're going to expand on that, in which we, instead of trying to pick one sequence out of a collection you get from an environment, we're going to look at the collection as a whole and try to figure out what that tells you about the population of organisms. There's a thing here about 454 pyrosequencing. Many of you learn, for example, in genetics or something like that, how sequencing reactions work. Typically, Sanger sequencing, so-called bideoxy sequencing, right? The kind of sequencing we're going into now, though, is high-throughput sequencing, where instead of doing one sequence in one tube, you're doing upwards of 500,000 or more sequences in a single tube. Really, really cool technology. 454 is where this started, and I've got a detailed description for how that works as an example of this sort of a thing. The reality is nobody does 454 sequencing anymore. That technology has already come and then gone. People use things like Illumina now, but they're all based on the same kind of things where you have to separate individual reactions into some kind of essentially silica plate where you've got individual wells and you're measuring stuff. Have a look at that if you wish. That's not really the point.